Wow, another one. So, casual conversation, cartoon, curriculum, very concise and relatively correct. As simple as possible, as soon as possible, A and P. So, chapter 22, third attempt at saying something. Please remember if you catch anything wrong or what I'm talking about, it's perhaps true because we're doing this real quick. Again, sort of casual, conversational, and kind of cartoony right here. So last time we were together, we were talking about the first line of defense, and we went through some of the things that you already know about the first line of defense. Now let's say something about the second line of defense. And most of this you know from some context also, perhaps chapter 5, when you were talking about tissues of the body, particularly areolar tissue. Because areolar tissue, you might remember, is sort of like jungle in that there's space there in which things can move around, and that makes a good place for white blood cells to be migrating through your body trying to find things. And then if there's going to be a battle, you've got a place where the battle plans work out. And then, of course, when it comes around to using chemicals to help fight this battle, here are some of the chemicals we're going to be talking about and the procedures too. So back up here at the top. Second line of defense. The second line of defense, of course, is going to be you broke through the wall, so to speak. You broke through the skin, you broke through the mucous membranes. Now in our analogy, this is like breaking through a castle wall. Now you're in the streets. And so these are the commoners doing their best to try to save the city, kind of a thing. We haven't called up the SWAT teams yet, the Navy SEALs, the Green Berets, the specialists. So just the common characters doing their job. One group that's definitely involved in this story are called phagocytes. And you remember that phago means to eat and cyte means cells. Here are three common phagocytes. I can't see it. There it is. Three common phagocytes that are part of our storyline. Stretch it out, bring it back. We've got macrophages, neutrophils, and eosinophils. Now, I printed them like this to remind you that these are by far the most efficient and effective. Coming in second place, let's say neutrophils, and way off in the bottom there, eosinophils. You might recall from chapter 18, we said that eosinophils in many ways are the cleanup crew after the big battle has happened. Eosinophils show up to clean up antigen-antibody complexes, and they can go after worms too. We said neutrophils are the ones that show up first, fast, and furious in large numbers, but unfortunately they're sacrificial because their weapons of defense, called defensins in general, aren't that great. Again, these are the main components of pus, but they do buy time so that monocytes can convert to macrophages, and the macrophages can then show up like the cavalry coming in. Unfortunately, it takes about 12 hours for them to show up in any significant numbers. For macrophages, there's both free and fixed macrophages. The free macrophages are wandering around. The fixed macrophages have a spot where they're attached. We've made the analogy many times before that macrophages are like junkyard dogs. And sometimes that dog is roaming around free, and sometimes they're in a fixed position. By silly analogy, let's say that you had a junkyard right here. Here's the gate. All kinds of stuff in here. There's an office, and you got a dog right here chained up at the gate. That says that nobody gets inside this gate except that dog has an opportunity to go after them. However, when the day is done, you close the gate and you let the dog free inside the fence. This now means that if somebody tries to jump the fence or get through the gate, that dog can go after them. So that macrophage can either be roaming around inside your body looking for trouble, or could be at a fixed position guarding one particular spot, or just doing its work in one particular spot. Move down here. Phagosome, phagolysosome. General story is, here is a phagocyte. Here is something that it wants to engulf. It comes up to the thing, wraps its loving arms, pseudopods around the thing, brings the thing inside of itself, and now that thing is inside what's called a phagosome. The phagosome is that bubble-like structure, that vesicle that formed around the engulfed object. So it's a phagosome. Some means body. Now, the next step in this story is going to be lysosomes, so-called suicide sacs inside the cell, now can come on over to the phagosome and attach to the outside of the phagosome. You might recall that this is sort of like soap bubbles coming together. And when the lysosomes attach to the phagosome, what you got there now is a phagolysosome. So we went from phagosome to phagolysosome over here. The bubbles will pop to the inside. They'll drop their digestive chemicals on top of that thing, whatever that thing happens to be, and that thing is gone. Well, hopefully it's gone. So that's the process of a phagocyte finding something, engulfing something, and then processing the something, hopefully stopping it. Of course, there's always the possibility that that something they got inside can't be digested. So we like to tell the story of alveolar macrophages. That is, macrophages that are found at the very end of your respiratory branching of your bronchial tree in the alveoli. If something gets down there like tar and it's recognized and then it's engulfed, the cell may try to destroy it, but unfortunately things such as tar and silicon can't be broken down easily by the chemicals in the lysosomes, and so therefore the chemical stays there. If the cell then goes after another one, it'll end up with another. And then if it goes after another one, pretty soon the thing can have itself all burdened up with all these chemicals inside. Bad news is this is not a truly functional macrophage anymore. If something really bad shows up over here and it should go after the thing, it's kind of hindered by the baggage that it's carrying around. This is one reason why inhaling any kind of silicon like rock dust or hairsprays that sometimes have silicon is a bad idea. It's a bad idea to inhale smoke, any kind of smoke, cigarette, etc., too very deeply. Those become materials that these macrophages can't easily destroy. And because of that, you've compromised the defense system down there inside the lungs and made yourself a clean target for pneumonia or something such as that. So phagosome, phagolysosome. Next thing here, adherence to surface carbohydrates, opsonization. It turns out that very often to get some kind of object out here, the macrophage or the phagosome has to grab it. It's not just wrapping their arms around it. Sometimes it has to grab it to be able to pull it inside. Well, sometimes these characters out here, such as a pathogen, might have something like a slime layer on the outside, makes it slippery, and in essence, the white blood cell can't grab the thing and pull it inside very, very easily. So this slime layer right here is a kind of a defense mechanism of some of these bacteria, etc., which, by the way, is one reason why you might think that as a kid, when something was smelly and slimy, it was kind of nasty. Well, that might have been a bit of a clue to you that on the microscopic level right here, yeah, there were some things in there that are commonly associated with bad news, disease and such. So the smell and the sliminess sometimes are sort of innate ways of knowing that something is not particularly good for you. Anywho, well, with this situation right here, this, say, bacterium is pretty well protected. So there's got to be a mechanism where you can defeat the slime. And that mechanism is called opsonization. Now, sometimes the phagocyte will grab a surface carbohydrate of the target right here and pull it in like a leash. Sometimes it has to add a handle to the object to make it easier to pull in. So there are things such as antibodies and things called complement proteins, antibodies and complement proteins that can be used to attach to these critters. And in doing so, in essence, they put a handle through the slime layer that gives this phagocyte something to grab so it can be pulled inside. And again, that process of putting that handle on the object is called opsonization. And again, it's one of the major, good points about antibodies and complement proteins. They don't kill the thing that they're working on. They usually just make it easier for white blood cells to kill them then or later. Next on our list right here, respiratory burst. This thing's kind of a wide open topic. Let's shrink it down a little bit. Respiratory burst very often refers to the use of free radicals to destroy structures. Now, it's called respiratory burst because very often oxygen is one of the components of the free radicals. Now, free radicals in general tend to be large molecules that with time may become ionized. They may end up putting on charges where they didn't normally have charges. So that free radical now becomes problematic. An example of this, or excuse me, an analogy for this would be, let's say you built a clock using steel gears. Let's say one of those steel gears became magnetized. Well, it won't move as easily as it used to, and so therefore it becomes problematic. So free radicals very often are problematic for living things. Well, that makes them a good weapon. And so therefore your body has the ability, your white blood cells have the ability to make these free radicals and use them almost like secret chemical weapons against the things that they're going after, against the pathogens, whatever it is. And dumping these things on the structure that needs to be destroyed is called a respiratory burst. Again, a reference to the oxygen content right there.